Hi, this is Daniel Palou explaining how vision works. First off, we need to know some brain anatomy. So I'd like to start with the thalamus, which is the part of the brain responsible for receiving and processing all sensory information except for smell. And the thalamus is right here. And inside the thalamus, we have the lateral geniculate nucleus, or the LGN, and it's the primary relay center for visual information received from the retina. And back here we have the occipital lobe, which is the visual processing center, and it also contains the visual cortex, which is responsible for processing visual information located in the occipital lobe. And up here, which is not shown, we have the parietal lobe, responsible for spatial vision, or determining whether we focus on objects closer to us or farther away from us. Light enters through the cornea, which is a clear covering that protects the eye and begins to focus the incoming light. The pupil is a small opening in the center of the eye where the light passes through. The lens focuses the incoming light on the retina. The iris is a colorized part of the eye that controls the size of the pupil by constricting or dilating in response to light intensity. And the iris surrounds the pupil, and it opens up in darkness and it closes up in bright light. The retina is a layer of tissue in the back of the eye that contains photoreceptor cells. And over here we have the optic nerve, which is a collection of millions of ganglion neurons that sends vast amounts of visual information to the brain using the thalamus. Here we have the foeva, which is the central point of the retina and is surrounded by and contains cone cells, which I will explain in the next slide. But first, visual accommodation is the process of changing the curvature of the lens to keep the light entering the eye focused on the retina. These rods and cones are the photoreceptor cells that I mentioned before, and the rods, the red ones, are visual neurons that specialize in detecting black, white, and gray colors, and they help us to see in dim light and with our peripheral vision. And cones are visual neurons that specialize in detecting fine details and colors, and they help us to see clearly in color and in bright light. Okay, these bipolar cells over here are important because they transfer information from the rods and cones to the ganglion cells. And the ganglion cells help us to recognize the visual images by creating an action potential. Okay, and I don't have a picture here, but feature detector neurons are specialized neurons that are located in the visual cortex that respond to the strength, angles, edges, and shapes and movements of visual stimulants. And all feature detector neurons work together at the same time. All these cells that I've mentioned before are essential in the process of phototransduction, which turns light into electrical signals that get sent to the brain and processed into visual information.